Welcome to our presentation. It's Christian Itin, Chairman and CEO of Autolus Therapeutics. I'll be introducing you to our company. We're focusing on next generation program T-cell therapies. We're a publicly listed company, so please look at the risk factors filed with the SEC. I'd like to start out with our lead programs. Key focus that we have is on acute lymphoblastic leukemia with our lead program called AUTO1. And second program is targeting diffuse large B-cell lymphoma, a program called AUTO3. Both programs, we expect significant steps during the course of the next 12 to 18 months. Obviously built on a unique set of technologies that I'll introduce you to as we're going through the presentation. Now, we do work on additional programs in clinical development. In addition to AUTO1 and AUTO3, we're also working on a T-cell lymphoma program with our product AUTO4 and a neuroblastoma targeting program called AUTO6. Moving to our lead program AUTO1, which is tailored for patients that have relapsed refractory acute lymphoblastic leukemia, and in fact, a focus on the adult patient population. We have approximately 3,000 patients between the US and Europe that are at the final stages of their disease, having failed high-dose chemotherapy, having failed transplant, as well as often the targeted therapies, blinatumomab or inotuzumab as well. So this is a very dire outlook for these patients. They usually have very limited amount of time to live left. We're also, when we think about the disease setting, it's a particularly challenging one because it's a disease that resides in the bone marrow. In fact, it's a disease that's incredibly aggressive. The cells have almost stem cell-like features, grow enormously rapidly. And at the same time, the patients, because they tend to be elderly, many of them, but also because they've experienced highly intense chemotherapy regimens, as well as obviously very aggressive regimens in the context of stem cell transplantation, tend to be in a very poor state. And so you also actually need to have an ability to combine this enormous amount of clinical activity with a good safety profile. And as we're dealing here with a cell-based therapy, we need to put pressure on that leukemia for a long period of time. We're looking at 18 months, maybe more, to actually get to a transformational outcome. So you need exceptional persistence. To combine all of these three features into a single product has been enormously challenging. And as a consequence, there's no CAR-T product currently approved for that indication. One of the key things that we did find as we're looking carefully into that space is that the original products did not behave in a physiological way, and as a consequence tended to get overactivated, producing lots of cytokine, leading to cytokine release syndrome, but also to extensive amount of neurotoxicity, which was a real challenge with the initial programs in the space and led to the discontinuation of some as well. So what we designed with AUTO1 is a product that actually can address the leukemic cells in a way that's very similar to a natural T-cell engagement, very rapid engagement, rapid kill, can actually dissociate again very rapidly and move on, attack the next cell. And having this type of a feature, which is the physiological path that T-cells are taking, allows us to actually get high levels of activity without actually driving the toxicity. And at the same time, because we actually have our T-cells in as good health as possible, very long persistence. Moving to our initial clinical experience, last update at the EHA in June, what we can show is that we have a very high level of CR rate, more than 80%. In fact, also the patients, all of them achieve molecular complete remission as well. And the event-free survival at 6 months is at 62%. And that comes with a good tolerability profile with no high-grade cytokine release syndrome and a limited amount of transient neurotoxicity. When we look at blinatumomab, which is the standard of care in this setting, we do see that we're at least twice as active as blinatumomab. If you look at molecular complete remissions, blina actually comes in at about 28% versus the more than 80% that we're seeing with AUTO1. The event-free survival for AUTO1 is twice as long or twice as significant, obviously, with compared to blina, and the overall adverse event profile is comparable to blinatumomab. So when we then look at durability of effect, this is an update. First, I'd like to highlight that The patients that actually have received this transplant after receiving Auto1 are the ones that are marked with a red star. So the majority of the patients have not received a second transplant. And actually what we're looking at is the single agent activity of Auto1 in these patients. As you can see, we have first patients that actually have crossed 18 months in molecular complete remission, which is exceptional. And we obviously think that we're starting to get to a place where we hopefully can see some of these patients getting into long-term remissions. Moving to the next steps with the program, obviously seeing an activity that is about twice as active as blinatumomab, we can drive this program forward in a single-arm study targeting 100 patients with relapsed refractory disease. We look at complete response rate as well as molecular complete response rate, event-free survival, and duration of response. We expect data, top-line data by the end of next year. Switching gears and talking about diffuse large B-cell lymphoma. This program is tailored to address one of the key challenges that we have seen in this space. On the one hand, we do see that the first generation of programs achieve complete remissions that are lasting. That is a first that we see in this field for third-line settings. But what we also do see is that about a third of these complete remissions that are initially induced are lost within a 3 to 6 months period. The reasons for the loss are twofold. Number one, about a third to up to a half of the patients lose the very structure that the CAR Ts were designed to bind. So it's antigen loss as the primary driver, first key driver for relapse. The second key driver for relapse is upregulation of PD-L1. About 2/3 of the patients at time of relapse have elevated levels of PD-L1 expressed on their lymphoma. So what we're doing with this program address these two key issues. First, having two different types of receptors in each of the CAR T cells, which gives us an ability to actually recognize the cell either through CD19 or through CD22. Each of those recognitions individually is sufficient to kill the target cell. So in other words, we're making it very difficult for a cell to make itself invisible because that cell has to lose simultaneously both CD19 and CD22, and that is obviously a very unlikely event. Secondly, what we do is we also provide or give a single dose of pembrolizumab as part of the preconditioning regimen to counteract the activity or the effect that PD-L1 upregulation can have on the lymphoma cell. So when we look at the outcome with this population, we do see a very good level of activity. Overall, we see about just shy of 50% CR rate. When we look at the patients at the recommended phase 2 dose, that CR rate is in the 60% range. And that obviously gives us a very strong starting point for this product. Now, when we look then into the durability of effect, what we have also seen and updated at ASCO is that actually we haven't seen any early relapses, which is obviously exactly what we were looking to prevent with the design that we're putting into this particular approach. Now, the good safety profile that we have seen does matter. And it does matter because the majority of patients with DLBCL are not treated where currently the CAR T therapies are actually offered in the US. The majority of patients are treated outside of academic centers. They're given in non-academic hospitals, in and outpatient setting, as well as in specialized oncology network clinics, again, in and outpatient settings. Currently, the patients that are being treated with CAR T are pretty much the inpatient segment of the centers of excellence of the academic centers, and that literally just allows you to reach about 10 to 20% of the total population that is in need of therapy. The good level of safety that we see, we believe, should allow us to actually get our therapy active and delivered across the entire settings of care. So this program is designed as a first-in-class CD19, CD22 program. It gives us a very unique design with the product. It gives us a very high level of complete remissions that appear to be sustained over time, and combine that with a very attractive safety profile. So next, T-cell lymphoma, quick view on AUTO4/5. These are programs that obviously are tackling a very challenging disease in its own right, because now it's the T-cells themselves that become malignant. And one of the challenges we have had across all therapeutic approaches that were tried in the space is to find an approach that is specific to the T cell lymphoma, and at the same time doesn't actually kill all the other T cells in the body. And so this is about a very unique approach to targeting, which we designed around very specific epitopes on the TCR beta chain that come in two different formats. And we basically were creating products corresponding to the TRBC1 and the other one to TRBC2. This program is very active, required a very sophisticated protein engineering approach to actually create the binders to be able to do that quite unusual type of targeting. And these programs are moving obviously in development. Auto4, as indicated, is in the clinic, and Auto5 is being prepared for a clinical start next year. When we then look quickly into the pipeline, I'd like to just briefly introduce you to kind of the technology that we're using. So the way we actually program our cells is actually not at the level of knocking in genes or knocking out genes, but what we do is we create protein modules that have an ability to change the phenotype of the behavior of the cell. Obviously, you already talked about chimeric antigen receptors as the way to target the respective tumor cell. And of course, getting those right, etc., is something that requires quite a lot of optimization, as you've seen with our Auto1 program. It is very important to get the targeting right. We then also have obviously a set of switches that allow us to control the activity, and a very significant amount of focus goes on to tackling the difficulty in the tumor microenvironment with a number of modules that can actually address that, including actually protein modules that can actually render the cells allogeneic as well. Now, just as a brief outlook, those are a number of the programs that we're currently working on. We have programs that are moving into the clinic later this year. This is a next-gen version for pediatric ALL in particular, called Auto1NG. We have programs that actually start to address the complexity of solid tumors. This is Auto6NG program, very excited about, which just gave an update at AACR, as we did also for Auto7 in prostate cancer. These are programs that combine several programming modules to actually change the environment within the solid tumor, we believe are absolutely essential to be able to crack those indications. So with that, just one final view on manufacturing. Obviously, we do work with the patient's own T cells. So this is an autologous approach that we're using. We're running this on a semi-automated setup. What you see on the right-hand side is the suite that we're doing the clinical manufacturing in. And then obviously, we're also building the commercial capabilities, which we're actually building in the US in Rockwell. So with that, I think we have come to the conclusion here. I appreciate your attention and looking forward to seeing you in person, hopefully not too far out. Thank you.